Today we'll start our next chapter, which is called Reproduction in Plants. Now, reproduction is basically the prime objective of all the forms of life. The sustenance of species depends upon reproduction. We are studying reproduction in plants, so first of all we should also know what plants are. Now, plants are multicellular autotrophic forms of life which are usually characterized by presence of vascular tissue. In lower plants, vascular tissue may be absent, such as in case of liverworts, there is no tissue. However, all plants, when we are talking about plants, what we mean to say is the liverworts, the bryophytes, the angiosperms, and the gymnosperms. So these are the different types of even pteridophytes. So we are talking about these particular autotrophic forms of life that make up the plant. Now, all plants are characterized by one very interesting feature, which is called alternation of generation, and we are going to start our chapter by understanding this, because later on this will be very helpful for us when we are studying higher plants, the angiosperms. Now, what is angiosperm? Angiosperms are they usually are unique to plants, but now recently they've been found. This particular phenomena has been found and discovered in a couple of fungi and slime molds. Now, slime molds are kind of fungi. Sometimes they are classified under protists. Now, what is alternation of generation? As the phrase itself points out, it is basically the oscillation of a form of life between two different types of generations. So, a plant in its life exists in two different types of multicellular forms. It exists as a diploid multicellular organism, and it also exists as a multicellular haploid organism during the same life in its life. Now, if you look at human beings, we are multicellular, but we are diploid. But we don't produce spores, so we are not sporophytes. Human beings have a multicellular life or a body which is diploid. We do not have a multicellular haploid life. We have a unicellular haploid type of a cell which we produce, which are the gametes, but we do not exhibit these two characteristics or these two multicellular bodies during our lives. But plants do. Now, in plants, the multicellular diploid body is called a sporophyte for the reason that it produces spores, and the multicellular haploid body produces gametes. Hence, that particular part of life or that particular multicellular body is called a gametophyte. The term sporo means spores, and the term phyte here means plant. So, sporophyte, the plant body that produces spores, and gametophyte, the plant body that produces gametes. Let us understand alternation of generation by taking an example, and here we are going to study the life cycle of moss like funaria. So here what we have is we have a moss. Let me just magnify it a bit so that we get a little bit closer. So here we have the life cycle of a moss, and we are going to start with the gametophytic phase, and I'm sure that all of you've seen this. We have studied this in class 9. Now, this is what a moss basically appears like, the photosynthetic part everybody's seen. Now, these photosynthetic parts, this particular part, this multicellular body here, is diploid, and on top of this diploid body we have got a sporophyte. Now, how is this sporophyte formed? We'll start studying it from here. So at the very beginning of the life, let's start the life cycle. Let's start to understand the life cycle of a moss from here, from this part, from the gametophytic phase. Now, we have a female gametophyte, which means the plant body that produces the female gametes, and the male gametophyte which produces the sperms. Now, there's a structure called antheridia present in the male gametophyte, and archegonia, which is a structure present in the female gametophyte. The antheridia produces sperms. These sperms are transferred to the female gametophyte, but where on the body? Into the archegonia. And what transfers these sperms? They are transported by the means of water, and that is why we call them, they are like very much dependent on rain for their propagation or presence of water or moisture. Right, now, once the sperms reach the archegonia, fertilization takes place, and I am sure you know that the gametes are haploid. The gametes are haploid cells. The haploid is N, and when they fertilize, they'll give rise to a diploid cell. Now, the diploid cell here, so this the diploid cell that we have here eventually then divides further by the process of mitosis, and mitosis will give rise to a diploid body. So we have a diploid zygote, a diploid embryonic form, and that diploid embryo then grows on top of the female gametophyte. That diploid structure grows on top. See here, this is that structure that grows. But the structure, this particular structure is diploid, right? I hope this is clear. Why is it diploid? And when the structure matures, it forms a structure called the capsule. So now here is the capsule. Inside of the capsule we have cells, the spore producing cells, and these spore producing cells are diploid because the plant body is diploid. So these cells are also diploid. How are they formed? The spore producing cells, they are formed by the mitosis, by the process of mitosis of the original cells of the present inside the or spore producing mother cells. So the spore producing mother cells are diploid, and these cells, they undergo meiosis or reductional division, and as they undergo meiosis they produce spores, and each spore mother cell will give rise to 4 spores. And when the spores are mature enough and when there is time, the capsule opens up and haploid spores are released into the environment. These haploid spores, when they reach the surface, they germinate. Because they are haploid, they will germinate and they'll give rise to haploid structures called protonema. You don't need to remember protonema. It appears like an algae, and slowly this algae, this filamentous structure protonema, eventually grows up into these 2 plant bodies. Now some of these protonemas will become female, some will become males, and these plant bodies are again haploid because they are formed by haploid spores. So what is interesting to know is that the gametophyte, the gamete producing body, is haploid because they are formed by spores, haploid spores. And these 2 gametophytes, when fertilization takes place, when the sperms are transferred from the male gametophyte to the female gametophyte, post fertilization we get a diploid spore producing body, which is the sporophyte, a diploid sporophyte. So in the life cycle of a moss, we can clearly see a sporophyte and a gametophyte. Now in the life cycle of the moss, for the most of during its lifespan, for the most of the period, the gametophytes find prevalence. They are the ones that are seen for most of the times, and sporophytes are short-lived. They appear for a very short period of time, and that's why the major generation in mosses is the gametophyte. So I hope you've understood what is the meaning of the term alternation of generation, and let us now proceed further to understanding reproduction in plants. And we will only study angiosperms at your level to understand reproduction. We will not study gymnosperms or pteridophytes or any other forms of plants. We'll study angiosperms. Now angiosperms are basically all the plants, they exhibit haplodiplontic life cycle. What do we mean by haplodiplontic life cycle? This means they have a multicellular haploid form, and they also exist as a diploid multicellular life form. So the same plant body will exhibit a haploid multicellular form and a diploid multicellular body at a certain, not together, but at certain periods of their life, like we saw in mosses. The life cycle starts with a gametophyte, and then slowly a sporophyte also develops in the same body. Right now, the dominant generation in angiosperms or flowering plants. Now I want you to first of all think of a flowering plant. The dominant generation is a sporophyte, which means the plant body, the most prominent plant body that we see, the one that we see with the leaves and the flowers, is actually the one that we see for most period of our life is a sporophyte, which means it produces spores. Now you must be wondering, no, I've seen plants with flowers which produces pollen grains, and pollen grains have got the gametes. So this plant should be a gametophyte. But now this is the point where you're going to learn something very new, very interesting. Now what is this pollen grain, and why is this plant not a gametophyte, is something that we will discover just now. So the plant body is a sporophyte, and we'll realize why. In order to do that, we will have to first of all look at the structures of the the reproductive parts of the plant. And today we will only delve into understanding the male reproductive part, the building unit of which is the stamen. Basically, the male reproductive part or component of the angiosperm is called the androecium. And androecium is made up of units called the stamen. We have two or more than two stamen in the flowers. Okay, there can be n number of stamen. This is a variable number. Now, once we understand, let us first of all start off with what the stamen is. Later on, we'll go to the term microsporangium and also the pollen grain. What is a stamen? Now, stamen is a male reproductive unit which is composed of two parts: the anther and the filament. So here we have this long structure. Here is the filament, and then we have the anther. Now, this particular part, the base of the filament, is connected either to the thalamus, which is also called the receptacle. This is basically the base of the flower. Okay, or this anther can also be inserted in. When I say inserted, it means attached to. An anther can be inserted in the thalamus, or it may be inserted into the petals of the flower. I hope you know what petals are, the colorful leaf-like structures. You may also find the stamens inserted onto the petals. Under certain circumstances, in some cases, the filament is also inserted in a structure called tepal. Now, I hope you remember from last year, a tepal is the unit of the whorl when you cannot distinguish between a sepal and a petal. For example, in morning glory, we have morning glory is that flower which is slightly purplish in color. In that, the lower part is green and the upper part is colorful violet. So this is neither a petal, because it appears like a petal on the top, but it appears like a sepal. It is photosynthetic at the base. We call this structure, it's not called a petal, it's not called a sepal, it's called a tepal. Now, in cases such as in morning glory, the filament is inserted in the tepal. Right. So I hope you know, I hope you've learnt or realized by now that the filament can be inserted on the thalamus, which is the base of the flower, or on the petals, or on the tepals, and in some cases maybe even on the sepals. So proceeding further, let us look at the other part of the male reproductive unit, the stamen. Let us now go to the anther. Now, anther is usually in angiosperms is bilobed. Now, what we mean by lobe is, let us magnify and try to understand what is this condition called the bilobed condition. Now, if you see in this particular anther, you can see two lobes. So this area, so I'm going to shade one of them. This is the first lobe, and let's use a green marker, and behind this, if you see this another one, this is the second lobe. You can see these lobe clearly if we take a transverse section. So if I cut this anther, I take a slice from here like this and put it under the microscope, or maybe if I just cut it like this, what will I see? I'll see this. So this is called a transverse section. So if you look at the transverse section carefully, what we will see, we can very clearly see the two lobes. So this is the first lobe here, this is the first lobe, and this is the second lobe. So that's why the anther in most of the angiosperm is bilobed. It's a bilobed anther. In some angiosperms and in some plants, you can also have a unilobed anther or a monolobed anther, right? But let us not go there. We'll just stick to the bilobed anther. So in most of the angiosperms, the anther is bilobed. And let's demagnify and go to the next point. Now, each lobe, I've already described to you what a lobe is. Now, each lobe, give me a moment, and here, let's fix it up. Now, each lobe here, so we see this is a lobe here. Now, what am I choosing? I have to choose a marker pen. So each lobe here, so this is one of the lobe. Each lobe is dithecous. Now, the term di means two, and thecous means wall. Now, each lobe is dithecous, which means each lobe has two walls. And let's get a little further and see where are those two walls. So you see, this is the first wall, because this is the wall of one of the chambers. We'll just come to what that chamber is. So this is the first theca, and this is the second theca. And each lobe is called dithecous, the first wall and the second wall is right here. So I hope you've understood the meaning of this particular term called dithecus. It's sometimes also called bithecus. Di and bi simply means two. Now both of these lobes are connected to each other by a tissue called the connective. Now how simpler can it get? Now let me use this green marker and let me show you where the connective is. So this structure here is the connective. So this part here is called the connective, and this is the connective right here. Connective connects the two lobes together. Also, the two lobes are separated from each other with by the presence of a long groove. I'm going to use my the pointer here, and you see this groove. So this long groove separates the two lobes. So two lobes are separated out, and now each lobe, let's magnify again, clear up the screen a bit. Now each lobe has chambers in it. Now if you look at this particular lobe, so we have one lobe here, one lobe here. Each lobe has two chambers. So can you see this first chamber is here, second chamber is here, this is the third chamber, this is the fourth chamber. So basically there are four rooms, four long tower like hollow rooms. So this is one room is just behind this, one room is here, one room is here, one room is here. And these rooms are not hollow, they are full of tissues, they are filled up by cells, and these four structures are called the sporangia. Now each one of this particular structure is called the sporangia. So we have got first sporangia, second, third and fourth, and that's why the bilobed anther is also called tetrasporangiate. So first sporangia is here, second sporangia is this, third is here, and fourth is here, and that's why the bilobed anther is also called tetra, which means four sporangiate. Now what is sporangia? I've told that these rooms are called sporangia, but sporangia actually means a place where spores will be formed. So inside of these sporangia, spores are formed. So how we are just getting closer to the idea why the plant that we see is a sporophyte. So at the moment the pollens have not been formed. We are moving into that direction, and we'll soon understand, or we'll soon be able to figure out why the plant body is the one that we see is usually considered a sporophyte. Alright, so once the pollen grains are ready inside of these sporangia at that particular time, just before the formation of pollen grains, these sporangia are not called sporangia, they are called pollen sacs. And these pollen sacs later in their life form pollen grains, and those pollen grains have to be released out. So for the pollen grains to release out, the pollen sacs split up. Tomorrow I'll show you a video where you can see the splitting, and this splitting is called dehiscence. So dehiscence can happen in many ways. In some cases the walls of the pollen sac simply develops pores. In some cases they develop flap or cap like structures, right? So usually the dehiscence or the rupturing of the pollen sac takes place longitudinally so that the pollen grains can be released. Now don't be confused. Remember that the sporangia we spoke about are the rooms. Inside the sporangia, spores are formed, and these spores will somehow give rise to pollen grains. Once the pollen grains are formed, the sporangia are not called sporangia, but they are called pollen sacs, and pollen sacs will release mature pollen grains. So I hope this much is clear to you, and now we'll move to the next slide and we will look at a couple of other terms. Now first thing that we just finished was the stamen. We understood what stamen is. We have understood what sporangium is. Now the spore producing tissue in the male reproductive organ, which is the anther. Pay attention to this. The spore producing tissue in the male reproductive organ, or the structure or the part, which is the sporangium here, which is the sporangium of the anther here, is called microsporangium. Microsporangium will, the sporangium here of the male is called microsporangium because it will give rise to male gametes later in life, and the male gametes are smaller. The similar structure, same kind of sporangia that we'll study later, which is found inside the ovary, is called megasporangium. But at the moment, why am I calling sporangium as microsporangium? Microsporangium means the male gamete producing sporangia. Yeah, the sporangium that belongs to the male reproductive organ. So this is the microsporangium. It is sporangium, but it's called micro because it will give rise to the male gametes, and male gametes are much smaller, much tinier than the female gametes. Right. So we have so far studied what a stamen is. We have understood the different parts of the anther. We've got into the details of it. We've also, I hope everybody knows what a pollen grain is, but we'll come to the technical details of it in just a little while. Now next, let us get in deeper into the structure of the anther. Now this is an immature anther at the top. Here we have an immature anther. Now the anther is made up of a couple of layers, and let us magnify and see those couple of layers. Let me just bring down. So this is the section of the anther, and inside of this there are a couple of structures. I hope you remember connective. So this particular part, connective, connects the two lobes. So we've got the first lobe here and the second lobe here, and if you wish to call this the first lobe, that is fine, and this is the second lobe. So there is no hard and fast rule about this. So the connective not only connects the two lobes together, also inside the connective we have got the vascular bundle, the xylem and the phloem. So the nutrient is brought into the anther through these particular vascular tissues. So connective has two roles. It connects the two lobes and also brings nourishment and all the necessary requirements for the tissue in the anther. Now remember, it's a reproductive tissue. The energy demands are very high, so the supply has to be really good and continuous. Now let us look at the other parts of the anther. The outermost layer of cells is called the epidermis. So the first layer of cell, the outermost one, is called the epidermis. The layer of cell just beneath the epidermis is called endothecium. Then we got the third layer of cell, which is called the middle layers, which means it is not made up of a single layer of cells, it is made up of multiple layers of cells, and that's why this third layer is called the middle layer. And inside of that we have got again a very thin layer of cells, which is called the tapetum, and inside the tapetum, so third is the middle layer, fourth is the tapetum, and inside the tapetum we've got the sporogenous tissue. Now epidermis, I hope you've all studied. Let us demagnify and go to slightly different area. Let's come here, and now what we are going to see is, let's demagnify just a little bit. Now here we are just going to take a little bit of the section of the whole anther, and we have magnified it here, and let us look at all of those four layers of cells, or basically five major zones of the anther. So the first zone, or the first layer of cell, the epidermis, is outside. You can see it's a single layer of cells, and its function is to protect or to cover, protect the anther. Similarly, we got endothecium. Endothecium also has the same protective function. Again, we've got the middle layers. Middle layers also have the same protective function. Along with the protective function, now they also have a slight bit of role in the formation of the pollen grains that you will learn tomorrow when I show you a very fantastic video. Anyways, now inside the middle layer, then we have got tapetum, and these are the tapetal cell. If you look at these cells with the nuclei and certain other organelles, these cells, they form the tapetum, or the tapetal layer of cells. Now the function of tapetum is to provide nourishment to the cells that lie inside. Where inside? Here, to the sporogenous tissue. Now the function of tapetum is to provide nourishment to the sporogenous tissue. Now what does this term sporogenous mean? Sporogenous. Now remember, genesis or genus simply means birth or the beginning. Birth or the beginning. And sporo simply means spore. So this tissue at exactly the center of the sporangium, microsporangium. Remember, this is a microsporangium. The entire thing is a microsporangium. And how many microsporangia we have got? We have got 4. Tetrasporangiate. Right. So the sporogenous tissue, which is exactly at the center, will give rise to spores. Now I can move further into this particular direction. We can learn a little bit about it today, and tomorrow maybe we'll have a little bit of revision. So here the sporogenous cells, let's go to the next slide and see these sporogenous cells in just a little moment. Let us just demagnify it a bit and come back to where we started from. So the sporogenous tissue is made up of cells. Now let me just pick up a marker. So these cells here, you see this particular cell here, these are the cells of this sporogenous tissue. Each of these cells is called a microspore mother cell or a pollen mother cell. Don't call it a microspore, please. Microspore mother cell or a pollen mother cell. So these are microspore mother cells. And the ploidy of microspore mother cell is diploid. The plant body that you see, the roots and the stem and the branches and the leaves and the flower that you see, these are all diploid. The cells are all diploid, and hence all the cells, even the cells on the inside, the microspore mother cells, are diploid. Now these diploid cells which are present in the sporogenous tissue, what happens to them? Now this is the pollen mother cell which is diploid. This undergoes meiosis. Now I hope everybody by now knows what meiosis is. Meiosis is a reductional division. Which means the ploidy is reduced from diploid to haploid, and this takes place in two stages, meiosis 1 and then we got something called as meiosis 2. The ploidy from diploid to haploid is achieved in meiosis 1, and then meiosis 2 simply doubles up the number of cells. So by the end of meiosis 1, let's look at this. So meiosis 1 has taken place. We have got a diploid cell, and by the end of meiosis 2 we got 2 haploid cells, but with double the number of, still there is double the content of DNA. So the ploidy is N, but this is 2C. If you don't remember this is okay, because I intend to once again do mitosis and meiosis sometime later in the class in details for better revision for people who've not understood it clearly. Even though we finished it last year, I hope you know the process. Anyways, so we've got 2 haploid cells now, and then meiosis 2 is more or less like mitosis, which means that each of these haploid cells will be again split up and they will divide. So at the end of meiosis, the gist of it all, what you must remember is, one pollen mother cell undergoes meiosis to give rise to 4 haploid microspores, and these 4 haploid cells, and each of this cell is haploid. So we got 4 haploid cells, and each of this cell is called a microspore. You see what has been formed now? A spore. Why do we call it a microspore? Because it is going to give rise to a male gamete. Male gametes are smaller in size compared to the female gametes, and hence this spore is called a microspore. Now, do you realize that the male reproductive organ, the anther, has given rise to spores? So this is the reason why the plant body is called a sporophyte. And what is the ploidy of this sporophyte? It is diploid, and this diploid sporophyte gives rise to haploid spores. Now the collection of these 4 spores is called a tetrad, and these 4 tetrads, eventually on the outside of these tetrads, these 4 cells, there is secretion of certain material. Now what is that material? There are 2 walls formed outside this microspore. Let's not don't look at this diagram at the moment. If you hurry, you will miss on certain things, certain very important things. So here let us just focus our attention to this. So we've got a microspore cell, and outside this, think about tapetum. What tapetum? The cells of the tapetum now start releasing certain contents. Now we get an inner layer which is made up of cellulose and pectin. These 2 components, both of them are carbohydrates, they form the inner wall of around the microspore. Each microspore develops an inner wall, and then there's this outer wall also which is secreted around them by the cells by the tapetum, and this outer layer is made up of a substance called sporopollenin. Now sporopollenin is made up of, this is really amazing to know, is made up of a pigment called carotene, the same pigment that gives your tomatoes and carrots the red color. The carotene is metabolized into a substance called sporopollenin. Sporopollenin forms the outer wall, and this outer wall is called the exine, and the inner wall, which is made up of cellulose and pectin, is called the intine. Now the intine and the exine both have got apertures in them, which means they have got tiny pores on them. Now I hope everybody's seen a pollen grain under a microscope in the past. Now, if you've looked carefully at the pollen grain, you might have found that there are certain perforations or holes, and these are the places from where the pollen tube arises. So this is also the place from where the cell that is lying on the inside can communicate with the external environment. Anyway, so we got the exine, which is made up of sporopollen, and entine, which is made up of cellulose and pectin. Now this compound called sporopollenin is extremely resistant to UV radiations, extremely resistant to desiccation. Desiccation is drying. Okay, so even if there is a lot of heat, whatever water is present inside the pollen will not easily evaporate, so it prevents desiccation. It is also extremely resistant to acids and large number of bases and variety of other chemicals. So exine provides extreme protection to the content on the inside, and entine also does the same. It also provides structural support because it has got cellulose, and there are a variety of other functions which we do not need to know at this particular moment. Now remember that this is a spore inside. Now let's go back to the moss. Okay, if you look at the moss, here, the spore, which is haploid, when it falls down, it germinates. What do we mean by germination? Spore is a single cell. It is a shell covering a single cell. When the conditions are conducive, the shell releases the cell that is on the inside, the haploid cell. This haploid cell undergoes mitosis, and mitosis will then make one more cell, and both of these cells will divide again. They'll form more cells, and eventually you get this multicellular structure which we call protonema, which you don't need to remember, which will grow into a multicellular plant body. So mitosis is taking place. What do we call this? This is called germination of spores. So the spores have germinated, and once the spore germinates, it's a different plant body. Right? Now, why did I tell you all of this? Because there is something that you now need to understand. So our pollen grain is not yet mature. It is still a spore carrying body, and we still have a sporophyte. Now you must be wondering, where is the gametophyte in the plant? Where is the gamete producing body of the plant? Now inside of this pollen grain, now this is the pollen grain. I'm not going to draw the exine and the entine. We have this microspore, which is a cell. This microspore, what is the ploidy of this microspore? It is haploid. Now this haploid spore, microspore on the inside, undergoes division via mitosis, and it forms, inside the pollen grain, it forms two cells. One cell is this and one cell is this, and one of the cells occupies a lesser space and the other cell occupies a larger space. So this cell, basically these are not cells, these are the two nuclei that have split up. Anyways, so now eventually they'll make up their own cells, they'll make up their own compartments. So now what we have here is, we have something called as the generative cell, or we also call it the generative nucleus. It's the same thing. Okay, generative nucleus or the generative cell, and then we've got the tube cell or the tube nucleus. So the tube nucleus is also called the vegetative cell. Vegetative cell. So the microspore divides, it splits into two, forming a vegetative cell or a tube nucleus or a tube cell, and the other cell is known as the generative cell or the generative nuclei. Generative cell or the generative nucleus. Now at this particular stage, what has happened? Similar to germination of spores, the microspore has also divided. And now the next life cycle has taken place. This vegetative cell or the tube nucleus, as the name suggests, will make the pollen tube during pollination. Once pollination has taken place, this will make the pollen tube. And the generative cell is actually the sperm. Now you see, the pollen grain holds the sperm. The pollen grain is also multicellular. Isn't it multicellular? Do you have a doubt? It is made up of two cells. It is not one, it is two cells. So the pollen grain is a multicellular structure which has given rise to the sperm. The pollen grain has given birth to the sperm. Why? Because it initially contained the microspore, and hence the pollen grain is the gametophyte, rather specifically the male gametophyte in angiosperms. So the pollen grain is the male gametophyte in angiosperms, and I hope The riddle has now been solved. Why? Where is the gametophyte? And the gametophyte has such a small body and a very short lifespan, and that is why the life cycle, the dominating generation of in angiosperms is the is the is not the gametophyte, is the sporophyte. You see, the dominant generation in angiosperms, flowering plants, is the sporophyte, because the plant produces the spore producing plant body is the one that exists for the most prominent period of the life, and the gametophyte is short lived, it has a very short life. So multicellular multicellular haploid plant body is a gametophyte. Now this is the pollen grain. Later on, when we come to fertilization, we'll see what happens. Now the last thing that I need to speak to you about is that the pollen grain usually is released at this stage from the anthers. The anthers, the bilobed anthers, they undergo dehiscence, the splitting up of the wall and the release of the pollen grains. So a large number of pollen grains are released at this particular stage, right? And this is called the two-celled stage. In some cases, however, the generative nucleus, even before pollination, even before the release of the pollen, let us take the tube nucleus away. In some plant species, the generative nucleus undergoes another mitotic division. If this undergoes a mitotic division, generative cell, it is already haploid. We have a haploid tube nucleus or a tube cell or a generative cell, vegetative cell. If the generative cell, only the generative cell undergoes mitotic division, how many cells we'll have? We'll have two cells again. And now what will be the ploidy of these two cells? Let's rub the original initial cell. And if what will be the ploidy? Because it's mitosis, so again both these cells will have the haploid number. Very few plants have pollen grains or release pollen grains in the three-celled stage. We call this the three-cell stage. First cell is here, which is a single vegetative cell, and then we got two generative cells or the generative nuclei. Right. So in very few cases, three-celled stage pollen grains are released. Now the two generative cells are always formed, but in most of the cases they are formed inside the pollen tube, which we'll see later in the videos that are coming up. I hope you've understood this much. Thank you so much for watching.